So let's talk about the characteristics that you can use to distinguish species, of which there are many, and then we'll finish up with an introduction to a couple different species concepts. So first of all, the kind of general question is, what is a species? Well, as soon as you figure that out, let everybody else know, because everybody's still trying to figure that out. Everybody has different definitions of what a species are. What we do know is that everybody's probably right, and you got to just make a decision about what is a species. But we do know specifically you can use these different characteristics to distinguish species. Now we're going to go through and talk about these characteristics, starting with the physical and morphological traits, and then finish up a little bit with these evolutionary and ecological factors that you can use. So typically, a species is going to refer to some group of organisms that maintains a distinct set of attributes in nature. You can kind of think about like true breeding genotypes that we talked about previously. But I want to note that single species can exist in two or more distinct populations that are evolving slowly into different species. So again, that touches on that idea of this continuum. So it depends on when you take the TARDIS down, at what point in time you're actually looking. If the separation or the time or the amount of separation is short, the populations can be very similar. However, if you could come back and look at that population, or go and look at that population thousands of years in advance, they may be very, very different at that point in time. So these characteristics that you use to define species depends on the type of organism that you're looking at. Now, first of all, what is most commonly used are the physical or morphological traits. Here, physical or morphological traits would be strikingly misleading. These are actually members of the same species. They're just different morphotypes. They are referred to, or in this case, you would have looked at them and said, "My God, they can't be the same." But if you look at their DNA, if you look at their behavior, if you look at ecological factors about them, they appear to be in the same species. Now, you can contrast that with a species that we talked about before, where they detected a cryptic species, which is a hidden species. These were frogs that looked exactly the same, the Hyla versicolor and Hyla chrysocoelus. They look the same, but one's diploid and one's tetraploid. And they have developed reproductive isolation, so they do not freely mate. They are a classic kind of species. So morphological or physical characteristics can be used, but they can be misleading. Here's some birds of different species. They actually, this is the western meadowlark and the eastern meadowlark. They're found throughout North America, and with this zone of contact or overlap in their distributions. They have very different mating calls, potentially as a consequence, in this zone of overlap, there's very little successful mating. So these appear to be on completely different, at this point in time, evolutionary trajectories, and very little mating going on or gene flow going on between these two species. So that brings us to this concept, this inability to interbreed as a way to define species. This is also known as reproductive isolation. So in this context, what we're talking about, or actually Dobzhansky, who is a very famous theoretical population geneticist, he proposed in the late 20s that species have this reproductive isolation from other species. Thus, they cannot successfully interbreed. And what he also broke out were these ideas of pre-zygotic isolating mechanisms and post-zygotic isolating mechanisms. Now, the pre-zygotic sounds like it is what it sounds like. This would actually prevent, or these types of mechanisms would prevent the formation of a zygote, whereas the post-zygotic isolating mechanisms prevent the development of a viable individual after fertilization. And we'll talk more about that viability and what that really mean by that. So in terms of these pre-zygotic isolating mechanisms, The first three listed here, habitat isolation, temporal, and sexual isolation, these mechanisms are primarily ecological or behavioral, right? So in terms of habitat, the two species never come into contact. So they're in different types of habitat. They're not gonna interbreed just because they're not in the close proximity. Temporal isolation, that could be something like having different spawning times. Corals in some places will spawn on different points, or different species will spawn at different times, and so you don't have that overlap for those broadcast spawners. So in terms of sexual isolation, that could be something as simple as that example of those meadowlarks. Early on, they develop this different type of call, and so they don't recognize as mating calls, and that can lead to that type of isolation, or prevent the formation of a zygote. Now, mechanical isolations mean that the parts just don't fit. Gametic isolation is the same idea. The gametes don't recognize each other, and therefore you don't get formation or fertilization. In terms of these post-zygotic isolating mechanisms, hybrid inviability, that basically means that you can get a fertilized egg, but it fails to develop, so past these early cell division stages. Hybrid sterility, everybody knows an example of that. That's a mule. These are animals that are still viable in the sense that they can survive, but they cannot reproduce. Now, hybrid breakdown is a little bit different in the sense that the F1 is typically viable and also fertile, but future generations are less fit. This is also known as hybrid vigor. So you get this kind of breakdown in the ability of these animals to reproduce in the future. Now, Ernst Mayr was another big or major proponent of some of the ideas of Dobzhansky, but he also utilized these ideas to develop the biological species concept, and that's the big one that we typically think of. That is grounded in this idea that if the animals can't interbreed, then they're different species. Now, there are some problems with using reproductive isolation to define species. And most of the time, this biological species concept is used to distinguish closely related species of animals and plants that reproduce sexually. So, as you can see here in this, we've got a whole sentence highlighting the problems with how do you use that to talk about asexual species, such as bacteria or fungi. You also have the problem that some of these species, because they don't overlap in terms of their habitat, in terms of their geographic distribution, you can't tell whether they actually might mate. So there's some Drosophila species that don't actually overlap, and they won't mate in the wild, but you put them in the lab and they do. So that can be problematic with defining it based on that concept. Then again, you can get some mating in the wild, but still maintain these very distinct characteristics that make you think that morphologically they are reproductively isolated. And then finally, this idea here, how do you check to see if extinct species were able to intermingle and to interbreed? Now, another thing that you can use instead of this interbreeding idea or the reproductive isolation idea are molecular features. And typically evolutionary biologists will utilize these, and more so nowadays than in the past. You can look at DNA sequences. You can look at gene order, simply trying to track how the gene order has changed over time. Chromosome structure and chromosome number are also used as characteristics. So not just the morphology, not just the external features, but also these internal features that lead to those morphological differences. This is a little bit more tenable for people that work with asexually reproducing organisms such as bacteria. In bacteria, you don't necessarily have a lot of morphology that you can use to define those species. So DNA sequences are a great fit for that. Now, however, one of the problems that arises, as with any kind of trait or character, is that it can be difficult to decide what a cutoff is. So, for example, if they are 10% different in the DNA sequence of cytochrome oxidase, does that make them a different species? Or does it have to be 30% different? Does it have to be as little as 1% different? Right? So there you have to make a decision about that. And there's been a recent rise in the use of barcoding to use that to define species. Again, people had to get together and decide what is going to be a cutoff for this. Now, one thing that this molecular features can be difficult would be for getting DNA out of fossils. That can be very time consuming and very difficult to get large pieces that you can use to define a species in that context. Now, there's two other kind of factors I wanted to touch on before we talk about the biological species concepts, and that would be this idea of these evolutionary relationships. These kind of relationships can be based on the fossil record. One of the problems of using evolutionary relationships to define species is that the fossil records are often times incomplete. And also, what do you do about soft-bodied animals? I worked on a series on slugs that had no bones, so they didn't leave any fossils. So that's problematic. You can look at evolutionary relationships based on DNA sequences, and that is a more common or a more tenable way to do it. Again, you still have the difficulty of getting DNA sequences out of these non-extant or non-living species anymore to try and fit them into that phylogeny overall. In addition, you can look at ecological factors. Also, hopefully, you guys might be thinking that you could use all these combined. You can use whatever information you can to actually piece together these kind of relationships to define species. Now, in terms of the ecological factors, you can think about where it would be. You could use more like behavioral traits, where different species of related birds might forage. What resources different bacterial species use, for example, the types of sugars, the types of vitamins, what kind of growth factors do they, or growth conditions do they thrive in. So different temperatures can define a species of bacteria. Think about archaeobacteria and the extremophiles. The problem with depending solely on ecological factors are that some species can show a great deal of variation in different types of growth conditions or resources that they might use. Now, I wanted to touch on just a few of the different types of species concepts that are out there. There are, last count, I think I've identified, not like I'm an expert on this, but just in doing some really short kind of searches through the literature, you can find at least 60 and up to 100 different species concepts that have been proposed in the literature. This is just an example of a couple of them. Here's the common one that was proposed in the 1920s by Mayer, and this is the biological species concept, and this is the one that you're often taught about. And that, again, relies on that concept of being able to interbreed to produce viable fertile offspring. There's also this concept of the evolutionary species concept. This one is where you're talking about these separately evolving lineages. Now, that is also linked to a phylogenetic species concept, where you can actually if you can differentiate a lineage by any kind of variation, i.e. any nucleotide differences, then it's a different species. There's the ecological species concept. That is using those ecological factors. So looking at a lineage, a species as a lineage or group of lineages that occupy some kind of ecological niche. Then there's finally the general lineage concept, And this one was proposed to really try and incorporate, or to be a more holistic approach, where you use multiple criteria. You use morphology, you look at reproductive isolation, you look at the DNA sequences, and you also incorporate the ecology of these different animals or different organisms. I'm sorry. So this was a more broad concept that you could apply to many different types of organisms.